the mechanism of protein synthesis. Mechanism that involves the formation of a protein in the cells. This process is also described as gene expression. We are already familiar with the term gene. A specific portion of the DNA molecule which is responsible for the expression of a particular hereditary trait has been called as the gene. The expression of that hereditary trait involves the synthesis of the necessary proteins. So, protein synthesis is described as gene expression. The genes have been classified into three types. Recons. The term recons is used to portions of the genes that are involved in genetic recombinations. And remember, these genetic recombinations occur during crossing over of the non-sister chromatids. So, those portions of the genes that are involved in genetic recombination, they are called recons. Mutons are portions of the genes that undergo mutation, that are affected by mutations. And the term cistrons, is used to describe portions of DNA that code for the synthesis of a specific protein molecule or a polypeptide. A protein molecule may have a single polypeptide chain, which means it is coded by one cistron. It may have two polypeptide chains for which two cistrons are necessary, more than two polycistrons, many cistrons are necessary. Protein synthesis or gene expression essentially has two major steps. Two major steps. One is called transcription. Transcription is the step in which a mRNA molecule is synthesized. A mRNA molecule is synthesized as a complementary chain to a cistron in the DNA. Cistron is the portion that carries the code. So transcription is the step in which a mRNA molecule is synthesized as a complementary chain to a specific region of the DNA, the region containing the code. The second major step is called translation. Translation is the step in which the newly synthesized mRNA associates with ribosome to synthesize a polypeptide chain. To synthesize a polypeptide chain. In this polypeptide chain, the sequence of amino acids will be decided by the sequence of nitrogen bases in the sequence of codons in the mRNA molecule. This sequence of codons in the mRNA molecule has been decided by DNA during transcription. So transcription and translation, these are the two major steps in gene expression or protein synthesis, and this idea is described as the central dogma of protein synthesis. Central dogma of protein synthesis. It is expressed like this: DNA by transcription, DNA by transcription synthesizes mRNA, and mRNA by translation synthesizes a polypeptide chain. DNA by transcription synthesizes the mRNA, and mRNA by translation synthesizes the polypeptide chain. This idea is known as the central dogma of protein synthesis.